Um, I have to start saying that it's a great day today. I think we are all remembering Pepe. I was thinking today the first time I met Pepe, and maybe that was in the mid-90s, 95, 96. I don't have the fixed date that I met him for the first time. Probably it was a meeting here in Spain, in the meeting in the Spanish Society of Microbiology. I have to say that from that year, we started to imagine collaborative work and to discuss about what we can do together. At that time, I came from Canada, from a postdoc that I was doing a lot of cell biology, analyzing the trafficking of Salmonella inside eukaryotic cell. And I was coming with this idea of testing antibodies, trying to get some insights on where Salmonella, in which kind of compartment was Salmonella. And I remember first talk with him, and we started to say, well, maybe we can complement very good each other because we have plenty of mutants in Seville. We can send you mutants. You can start to play with them in your epithelial cells and in your fibroblasts, and that was the case. I mean, we started to match very, very well, and we enjoy a lot. And for me, this is a very special day. So, as the title said, I'm going to talk about a small story. But the smallest story, but it's an example of how Pepe helped. I think I'm totally sure not only my group, but also many groups, because he had a huge collection of strains, and he was always open to, of course, to share this great material that he was generating in his lab in Seville. So this is just one example of many that probably you will have in mind that I hope you can see how this was much productive and helpful for us. So the story starts with a paper that Pepe published in 2001 in the journal Genetics. I think one of his favorite journals, right? Because this is the American Society of Genetics. And it was a paper that he described, as the title said, genetic mapping by duplication segregation in Salmonella enterica. So in this paper, in 2001, he was proposing a rapid method to map any mutation in the genome. And the idea in the paper was that he was generating duplications in the genome of Salmonella that were carrying a duplication with known endpoints. So he generated a collection of strains that, as you can see in this diagram, you can rapidly map any marker that you have, because if the marker was located in the duplicated area versus the case that was outside the area, doing the segregation of that marker, immediately you know if the marker was inside or not in that region of the genome. If the marker was outside, all the segregants were, let's say, if the marker is a tetracycline resistant marker, the marker was in both population of segregants. If the marker was inside, only half of the segregant will have the resistance. So this was great paper. I think I enjoy a lot reading it. And he demonstrated in the paper, this is just one example, that in this case, using some of these strains that were covering the entire genome, if this, what I was just saying before, in that cases in which you have segregants with the resistance, that means that they were located in a specific region of the genome. So what is the connection of this study with us? Is because we were at that time, and we continue working on peptidoglycans. So this is apparently very far from what the mapping strategy that Pepe published, right? So we are interested in the peptidoglycan, and why we are interested in this macromolecule is because it's essential, as you know, for the cell integrity and for the shape, define the shape of bacteria, and also because it's target of many antibiotics. There is a cytosolic route in sequential addition of amino acid to a sugar, that is an activity sugar, and addition of another sugar, and this molecule that is named DP2 is going to be flipped to the periplums, and is going to be incorporated in the peptidoglycan by two reactions. One is a transglycosylation reaction, in which the sugar is linked to the nascent peptidoglycan chain, and another reaction that is the transpeptidation reaction. So there is two coupled reactions. Not always the transpeptidation occurs, but in the case of the transglycosylation, as you can imagine, is essential to add new material to the peptidoglycan. So we are in the field of the cell wall and peptidoglycan. There are two types of penicillin-binding proteins that are going to do the transpeptidation reaction. One of these families is a bifunctional PBP that is able to do the transpeptidation and the transglycosylation reaction. And another one is called B family or monofunctional BPBPs that perform only the transpeptidation reaction. These PBPs are able to bind beta-lactams because the transpeptidation reaction mimics the structure. The beta-lactams mimics the structure of the diala, that is the terminal amino acid in the peptide side chains. They need the collaboration of a protein that is doing the transglycosylation reaction. So they work by pairs, not only a single one. So it's curious that these BPBPs, these monofunctional PBPs, are essential for morphogenesis. In a rod-shaped bacterium like Salmonella, you have two processes. One is the cell elongation, and the other one is the cell division. So now you can see I put here in blue PBP2 and PBP3, because this just what you can find in the microbiology books. E. coli, that has been the model organism in which much has been done regarding morphogenesis of bacteria, has these two PBPs working together with this transglycosylase, in the case of PBP2 with ROD-A, in the case of PBP3 with FTW. As you can also imagine, these PBPs are essential. If you knock out this, it's not possible to knock out this gene in E. coli, and also they are the main target of the bacteriolytic beta-lactams. Okay? So what is interesting also to know is that the PBP3, being an essential PBP, is also located in a gene cluster that is called division cell wall cluster, in which is located just mostly in the middle of the cluster, and there is a lot of genes that are downstream that are all essential. Many of these genes encode for the enzymes that are going to contribute to the synthesis of the precursor molecule of the peptidoglycan, and some of them, as these FTS genes at the end, are also proteins involved in cell division. So as you can imagine, this is a very tricky area of the genome. You cannot manipulate that because every attempt to knock out some gene that is in the middle is going to be catastrophic, and of course you are not going to get the mutant. Okay? In fact, this DCW cluster is intensively studied now. There's a recent paper in Nature Microbiology that shows that this cluster is very ancient and has been acquired by bacteria very early in the evolution of the bacteria domain. So, as I said before, we are interested in the peptidoglycan, and we are also interested in the intracellular lifestyle. So we want to know if bacteria, Salmonella in this case, with these two different lifestyles, is doing the same kind of peptidoglycan outside than inside. So why we suppose that must be any difference inside is because inside there are receptors that are going to recognize peptidoglycan fragments. So there should be some selective pressure to do something different here, okay? So concerning PBP2 and 3, we were surprised that when analyzing the genome of Salmonella enterica, we found FTSI, this gene encoding PBP3 in the DCW cluster, but we found another gene mapping elsewhere in the genome encoding for a predicted PBP that show high identity to PBP3. We named this gene at the beginning PBP3 star, and we were surprised because that gene was absent in E. coli. As you know, everything that has Salmonella is not in E. coli. You suspect that maybe there should be something related to the infection. So we started to be interested in this PBP3 star protein, and then this postdoc, Sonia, in my lab, figured out that this gene was mapping as well, but now the question was how to demonstrate that this PBP, that from now on we call PBP3 salt, whether this PBP3 salt was involved in cell division or not. That was a very intriguing question. We were, for the first time, saying that maybe one bacteria is using two different PBPs to do the septum, the cell division process. So what Sonia did was the easy experiment. And the easy experiment was, well, let's take these two PVPs encoding genes, and we are going to try to complement in an E. coli strain cell division. We have in a freezer an E. coli that I could recover many years, was in the freezer from my PhD, and we were able to find that mutant. And that was very insightful at the beginning, because this mutant is a PVP3-TS mutant. Can divide perfectly at 30 degrees, but at 42 degrees cannot divide. And the phenotype you see in the microscope are long filaments. Long filaments because bacteria continue growing without doing the septum. So you can see here, doing the standard assay and looking at 42 degrees, this bacteria in the empty vector with and without inducer filaments. When we express PVP3 of Salmonella typhimurion in this E. coli, now we got complementation. The bacteria was capable of doing division. However, we were not very happy seeing that the PVP3 salt, the other PVP with the control that was rightly produced, was unable to complement that phenotype. So one day Sonia decided to repeat the experiment by just changing the pH. Why changing the pH? Because we were discussing that Salmonella during the infection is living in a compartment that is acidic. And if PVP3 salt is not in E. coli, maybe it's because it's used by the pathogen inside the eukaryotic cell. And when she repeated the experiment, now at low pH, now she could recover perfect rod cells. So this was the first proof in an heterologous host as E. coli that PVP3 salt was able to do by itself, alone, independently, the cell division process. So now we have to move to Salmonella. Here is where Pepe really boosts our project. And this was very simple. We have to knock out PVP3 because we have two enzymes doing cell division, and we need mutant for each of them, because until we don't have those mutants, we are not going to be sure that really they can do the job by themselves. So this is what Sonia did at that time, and was using one of those strains from the collection, now having duplicate the region of the genome where is the DCW cluster. So what Sonia did was just to knock out one of the two alleles of the FTSI. Remember that everything downstream this gene is essential. So she knocked out, and then after that, what she did was to remove the antibiotic cassette because we didn't want any polar effect downstream. And now in this, sorry, and oh, I want to go back. And now in this strain, in this metadiploid strain, she did many attempts in neutral pH, no way to get segregation, but when repeating an acid pH, she was able to get segregants that in one case have the wild-type allele, and in another case has the null allele in PVP3. When the day she told me, I think I have it, I couldn't believe it. Because, I mean, saying that you have a mutant without PVP3, that was like going against the dogma, right? Against the microbiology book. And I said, do a Western, please. No, I have the PCR, do the Western, please. And then she did the Western. And I have this beautiful Western just in my office saying that, yes, in Salmonella, you can have a mutant that has no PVP3 and can divide and live perfectly. But guess now what happened with this mutant? We have knocked out PVP3. PVP3 works in neutral pH. And now we have a mutant that cannot grow in neutral pH because it's unable to divide a neutral pH. So this mutant now needs acid pH to grow and divide in the lab. So we have an acid pH mutant. Sonia has been doing many mutants, and we have now a mutant that is deficient in PVP2, because with PVP2 the story repeats. Salmonella has a duplicated PVP2, has 2 and 2 sal, and now we have a mutant that has not 2, not 3. And now you can see this mutant. If you put the mutant in neutral pH, the medium that everyone uses, this mutant is crazy, cannot do peptidoglycan, as you can see, explode, as the same as being in contact with a beta-lactam antibiotic. However, in this strain, you start to play with the pH and say, hey, you are going to be like being in the phagosomal compartment, and you reach this 4.6. Now you have beautiful rod bacteria. So this is what we have now, and this is a material that is very great to play with that, to analyze the peptidoglycan structure, to analyze many things related to peptidoglycan metabolism in these conditions that mimic the intracellular environment. What we have done also with this mutant now, we have generated the wild type, a mutant that is deficient in 3, and a mutant that is deficient in 3 SAL. So this is the value of the strain of Pepe. Without that strain, we were not able to get this triad of strains. With these isogenic strains, having 3 SAL, having both to divide, now you can play, do cell biology assays, demonstrate that in the absence of PEP3, 3 SAL is capable of promoting division once bacteria are inside the infected cells. And we are also something that is very important to do biochemistry. There is a lot of information concerning proteins that interact with PEP3, PVP2, and all the morphogenetic complex during elongation and division. Indeed, these complexes are known as elongasome and divisome complexes, where many proteins are supposed to interact there. But there is no biochemical evidence regarding the stoichiometry of these proteins in the complex. And now, this is one example with the triad that is wild type, deficient in 2, and deficient in 2 SAL, but we have equivalent data with the 3 strains concerning 3 and 3 SAL. Now you can do purify membrane extras and do pull-down experiments and demonstrate that in these strains, now you can do biochemistry, and now you can pull down complex in one strain, in this one, but not in the other, that is missing unspecific PVP. So unless having this isogenic delta 2 SAL, or delta 3 SAL, we were not unable to really confirm the presence of these specific complexes. One of the conclusions of this study is that apparently the PVPs are able to assemble in independent complexes to do the morphogenesis process. I have also to say that this mutant has been very useful. As also Stan mentioned, I mean, there are a lot of carriage of Salmonella, not only with type in humans, but also many serobars that, depending the host, can be in a symptomatic stage. And this is a really big problem, not only in human medicine, but also in livestock, in animal health. And this is because Salmonella has that potential to be asymptomatic. So in a recent paper, we just tested, well, if these PVPs are doing something in the intracellular environment, and there is a lot of relapse rates in Salmonella infections, maybe we can do the experiment in mice in which if we treat with an antibiotic like ceftriaxone, we can see that if we challenge with Salmonella, these mice are happy. However, if you remove the drug and you wait a few days, these mice become ill again. So that means that you can really test in mice the relapse phenomena. And now you can put in this system the mutant that is defective in 3 and 3 SAL. And one surprise that we found is that when using a mutant that is defective in PVP3 SAL, remember this is the Salmonella specific PVP, now these mutants, even after removing the drug, are happy again. So that means that this PVP, what it's doing in the intracellular environment, is going to be crucial to Salmonella to persist in that environment and probably to be responsible or contribute to the relapse that is known in a clinical setting. Last but not least, and it's also something that we are working very hard now, is that we want to block specifically the PVPs that are used in the intracellular environment. We have found, after a screening with some libraries, small libraries, but we were lucky to find one beta-lactam antibiotic, that is this cefotiam, that shows higher affinity for 3 SAL than for PVP3. So why we know that is higher affinity to one PVP to the other is that we can look at the microscope, and now with this delta 3 and delta 3 SAL have the evidence that the antibiotic at specific concentration can induce filamentation in one case but not in the other. So this is a direct proof that the antibiotic is going better to one PVP versus the other. So this is something that, as you can see, we have diverged, we have branched with many projects, and all has been thanks to the capacity of having these unique strains, because it's something that we cannot reproduce in E. coli, and this is something that Salmonella has in a very peculiar way. And now we can address many questions thanks to the strain and that paper and that collection of strains that Pepe generated. And I guess he generated hundreds or probably thousands of strains all over the world, and were made here in Seville. And this is, as I mentioned, wanted to exemplify how good has been for many investigators the work that Pepe did. Thank you very much. That was my time.